I'm John Cooper. I'm consultant pathologist to Jersey Wildlife Preservation Trust, which is in the British Channel Isles, just off the coast of France in Europe. I'd like first of all to acknowledge the support of my veterinary colleagues at Jersey, Tony Allchurch and Chris Dutton, and also those who've permitted me to use their slides during this presentation, Keith Lawrence, John Harshbarger, Richard Norman and Peter Holt. If we could have the first slide, please. My subject today is skin diseases of reptiles, and the first slide reminds you that the class Reptilia is one of the classes of the chordates, and that there are 4 orders of reptiles which are listed on the slide. But I should be concentrating on snakes and lizards and the chilonians, the tortoises, terrapins and turtles. The first point that we have to make when thinking about the skin of reptiles, and particularly the diseases, is the normal structure of that skin. Because if we don't understand normality, then obviously our understanding of pathology and how to deal with pathological lesions is very limited. The main feature of the skin of reptiles is the scalation. Here we have a lizard, a Uromastix lizard, for example, that is adapted to life in the desert, and you can see the scalation, different types of scales, on different parts of the body here. The scales, of course, are made of keratin, and they are impermeable in terms of organisms, of gases and so on, and they play a very important part in the protection of the integument of reptiles. This low-power scanning electron micrograph, for example, of the skin of a different sort of lizard, a skink, illustrates the overlapping shields, the scales there. There are no skin glands, certainly not here. Some reptiles have skin glands associated with the cloaca, sometimes in the neck region, but certainly not on the body. So these are protective, these scales. They're protective against physical insults. They protect against desiccation, one of the reasons why the reptiles have been so successful in gaining mastery of the land. And as I've said, they are impermeable in terms of other possible insults also. Right, when we look at the different types of reptile, though, we do have some differences. This is a giant land tortoise on the island of Zanzibar in the Indian Ocean, and here quite clearly a reptile, quite clearly pronounced scalation, but nevertheless a very different morphology. What we have here, of course, is the characteristic bony box of the carapace and the plastron here surrounding the body, and at first sight this would not appear, here's the caudal view, this would not appear to fit the standard pattern for a reptile. But in fact it does. What we have here is a modification of the pattern I described before. The body is still essentially covered by keratinized shields. You've got them here on the carapace and on the limbs. And the bony box, as I'm describing it, the carapace is a modification of the vertebral column and the rib cage, and then is covered with, first of all, periosteum, and then with epithelium, which gives rise to the keratinized scales, and the same applies on the ventral surface. If we move on from that and look at a histological section of a young chilonian, we can see that we've got bone, young growing bone here. We've got the stratum corneum with keratin on the outside here, and we've got a dermis that's very richly endowed with collagen here. So the basic structure is the same, even though tortoises, turtles and terrapins appear to be rather different. Let's then move on from there and think about skin lesions in reptiles. Skin lesions in reptiles, as indeed in any other species, can be broadly divided into those that are infectious and those that are non-infectious. But as I shall explain, there's sometimes an overlap between the two. And then in terms of the sort of skin responses we see in the class Reptilia, there are 3 basic ones. First of all, cellular infiltration, inflammatory cells, for example. Secondly, degeneration, various types of degeneration. And thirdly, proliferation, which is either reactive or it may be neoplastic, which again I shall refer to later on. So those are our basic responses, and I hope that pattern will become obvious in some of the examples that I give you. Let's move on then and look at some of the diseases of reptiles, skin disease of reptiles, and particularly the pathology of those skin diseases. I come back to my division between infectious and non-infectious diseases, and I'd like first of all to look at non-infectious, because particularly in captivity, non-infectious, particularly physical factors, are undoubtedly the most important. Sometimes the traumatic injury may be very minor and indeed physiological rather than pathological. This leopard tortoise, Testudo pardalis, for example, is interesting because the normal appearance of the shields, the scutes here, has been lost on either side. It has a rather dull, matte appearance and looks as if there's been some damage, possibly even erosion of this area. In fact, as I say, this is physiological, this is quite normal, this is a female tortoise, and this damage has been caused by repeated mating. Having said that, and this is one of the key features in the investigation of diseases of reptiles, close examination, good observation are vital, not just to the clinician, but to the pathologist as well. So if we in fact look rather more closely at that particular tortoise, we notice that there are in fact lesions, quite apart from the what I've described associated with copulation. There are in fact some pitted lesions on the scutes here, where the keratin has become pitted, and in fact these are little foci where infection during the rainy season, where gram-negative bacteria in particular have been able to get in, are beginning to underrun the shields and starting an infection, the equivalent of a cellulitis, but rather different since this is a reptile and the anatomy is rather different. We see many traumatic skin lesions in reptiles that are associated with management. Here, for example, a water dragon, we see a severe chronic lesion on the rostrum. You can see that there's been fibrosis and contraction and so on, and this has been associated with the lizard rubbing its head along the glass on the edge of the vivarium. But this sort of thing can extend, and I've seen bacterial infections of the maxilla and mandible, and indeed even osteomyelitis result from this sort of lesion. So management is very, very important, and we have to bear this in mind whenever we're looking at skin lesions. An example here is where one has competition and aggression between individuals. This is a freshwater turtle, or as we call them in Europe, terrapins, and this one has been in a fight. It's had part of its forelimb amputated by another terrapin, and now there's quite a severe, indeed a gangrenous dermatitis that's been has started there, with a fair number of gram-negative, with a fair number of anaerobic organisms, a mixed flora in fact responsible for this infection. So we have to bear in mind that, that these sort of management factors are very important when these animals are kept in captivity. What we mustn't forget is that certain changes in reptiles are normal, even though they might appear to be pathological. For example, autotomy, the ability of many lizards to shed, to lose their tail, and then to regenerate it, can result in quite an unpleasant lesion where the tail has been shed. This is one such shed tail from a gecko, and where the tail has left the body, there would be bleeding and apparently a skin lesion. This ability of many lizards to regenerate their tail is an important part of their behavior patterns and their survival, and is well recognized in many ways. For example, the British Association of Plastic Surgery has recognized this with the inclusion of a lizard in its coat of arms, a reminder to those involved in plastic surgery of the ability of some non-human species to do things that we're not yet able to do. So it's something we have to bear in mind in our differential diagnoses. This is quite normal in certain species, and we should be aware of it and of its significance. If we move on from there, there's a number of other physical, non-infectious factors that can cause skin disease in reptiles. Burns are one of them. Reptiles are burnt in the wild, free-living reptiles, during the course of grass fires or fires perhaps in someone's garden where a pet tortoise, a turtle, is exposed to it. But in captivity they are particularly prevalent, and here is a very severe example from Kenya, from a collection in Kenya, where a common green iguana has developed a very severe, not just a skin burn, but penetrating right through the peritoneal cavity as a result of sitting on its heater. And these sort of lesions are common in captive reptiles, and the first presenting sign is often what appears to be a fairly superficial skin lesion. An early burn in a reptile, in this case in a young python, on a biopsy shows characteristic signs. You've got here, this is an acute lesion, you've got a mixed inflammatory infiltrate here, which will be a mixture, predominantly heterophils up here, but a mixture of heterophils, granulocytes, and mononuclear cells here. Quite a lot of edema and disruption of the normal architecture. You see how the epithelium is being lifted off. So that is an early burn lesion in a reptile, and this is almost a pathognomonic lesion if you're looking at a biopsy or a post-mortem sample. Later on, burns in reptiles show characteristic scarring and fibrosis, and you can see this here in this snake. This is a burn that occurred 2 months earlier, and you can see the contraction and the changes in the appearance that are quite characteristic of a healing process. If a secondary infection doesn't supervene, and in burns in reptiles, gram-negative organisms such as Pseudomonas and Aeromonas are particular opportunistic infections of burns. If a burn doesn't become infected, then it should heal satisfactorily, as you see here. What is important to remember, though, is that there has been quite severe disruption here of the architecture as a result of the healing. If we look at a scanning electron micrograph, going back to my iguana, the earlier one, you can see that the normal scalation has been very disrupted and is actually missing in many areas. So although the wound has healed, it's not a perfect heal, and this will result in difficulties in skin shedding and sloughing, dysectisis, which I shall be talking about a bit later on in this lecture. So burns are certainly high on our list of differential diagnoses in terms of non-infectious lesions causing skin disease, non-infectious skin lesions in reptiles. There's a variety of nutritional factors also that can cause skin disease or contribute to it in reptiles. In the time available, I'm just giving 1 or 2 examples. Some of these can be specific ones. This, for example, is a vitamin A deficiency in a lizard, a colony of laboratory-maintained lizards, and it's a section, as you can see, through the eye. Here's the eye, completely closed. There's hyperkeratinization and parakeratosis here on the eyelids, and a number of other changes associated with this vitamin deficiency. These ocular ones were particularly pronounced, but there were others in terms of skin lesions elsewhere on the body, on the trunk, and on the limbs. More generalized nutritional deficiencies can also cause or predispose to skin lesions. For example, here we have a post-mortem specimen, an emaciated young Nile crocodile from a crocodile farm in Central Africa. Very poor condition, as you can see, wasted away. It's very important, whether these are clinical cases or post-mortem ones, to examine them carefully for skin lesions. And indeed, there are skin lesions here being pointed out just there, and there's several others as well in the rami of the jaws, etc. And so a generally debilitated reptile will very frequently present with skin lesions. These very rapidly become secondarily infected, and this fits very much what was the term the maladaptation syndrome, which was coined by Cowan here in the United States back in the 60s, and which is still a very useful method, a very useful system to follow in terms of the cycle of events when a reptile is in poor condition and a number of sequelae follow. Right, let's move on now then and look at some of the infectious diseases of the skin of reptiles. This is a general list. I'm not suggesting that all of these are necessarily important in infectious skin diseases of reptiles, but most of them can play a part in some way or another. We don't know about the organisms that would sit in the middle here, such things as mycoplasmas, etc. They've certainly been implicated in diseases of reptiles, but not yet in skin diseases of them. As far as the infectious causes of skin disease are concerned, the parasites, the metazoan parasites, are probably the most spectacular and the most frequently seen, particularly amongst those of us who work with free-living reptiles as well as those in captivity. This is the common snake mite. This is Ophionicis natricus, which is very common on many colubrid snakes, but can also, particularly in captivity, attack a range of reptiles, including on occasion lizards. This is an important parasite because not only does it suck blood, and large numbers of them can cause death due to anemia and associated changes, but also the parasite can transmit bacteria. An Aeromonas hydrophila, the gram-negative organism I referred to earlier, which is such an important potential pathogen of reptiles, has been shown experimentally to be transmissible via this mite. The actual lesions produced by the mite are generally very small, just the little puncture wounds through which it sucks blood, but the consequences of infection can be quite considerable. It's worth mentioning that other species of mite are found on reptiles, and again certainly can cause an effect in terms of their blood sucking and occasionally produce skin lesions or low-grade dermatitis. One important point that's worth mentioning is that some species, particularly of geckos, and you have a gecko here, this is a tropical gecko in Tanzania, East Africa, show mite infestations where you get the impression there's a predilection for certain areas of the body. And that is indeed the case, but it's not a predilection on the part of the mites per se. Behind the axillae of certain species of gecko, there are areas which are called mite pockets, and these are areas in which the keratin and indeed the epidermis are thinner than elsewhere in the body. And it's been suggested in a series of scientific papers that this is a strategy for concentrating the ectoparasites in one area rather than they're covering the rest of the body. There's some debate about that, and of course the other point here is that this is an area in which the lizard doesn't find it easy to remove the parasites. But be that as it may, congregations of ectoparasites in certain areas may be related to the anatomy, particularly the microscopic anatomy of the lizard, and that may have consequences in terms of the sort of lesions that they can produce. There are various other types of metazoan parasite that can be associated with skin lesions in reptiles. There's a number of skin-penetrating helminths, nematodes, for example, Callicephalus, which is the reptile hookworm, and the larval stage of that can penetrate the skin. Callicephalus is a parasite that has an alternation of generations, so it can live in a free living state in the absence of a host for long periods of time, and then when hosts are available, the larvae will penetrate the skin and produce skin lesions as they track through. This, on the other hand, is a phylloid worm. We don't know the species. It was one under the skin of a king snake and was only associated with a certain amount of hyperkeratinization on the surface and probably not of great significance, but nevertheless quite a spectacular case and an interesting one for teaching and comparative purposes. Right, the most common or the most frequently seen infectious skin diseases of reptiles are undoubtedly those caused by bacteria, and in particular abscesses. And these are seen in free-living, free-ranging reptiles as well as captive ones. Here we have a flat-necked chameleon in East Africa, one that I had literally just picked off a tree, and it has a small yellow raised lesion on the mandible, and this proved to be, on aspiration and cytology and bacteriology, proved to be an abscess associated with gram-negative bacteria. So these things occur in the wild as well as in captivity. But in captivity, abscesses are very prevalent. Here, for example, we have a green iguana which had multiple abscesses, but this one on the base of the tail illustrates nicely the sort of lesions, raised, firm, the disappearance of scalation on the surface, and in this case you've got erosion and probably early ulceration occurring. These are almost invariably bacterial in terms of their etiology. Sometimes we believe the bacteria are introduced from the outside, perhaps through a puncture wound, perhaps even through an injection or ectoparasites or trauma. Other times, particularly when they're multiple, we believe it's hematogenous spread of bacteria. When these lesions are removed, as they should be, excised in toto in terms of their treatment, If we bisect them, and that's what I've done here with this one from the iguana, we can see the overlying skin here. We can see that there is a well-marked connective tissue capsule. We can also see, I hope, that there's lamination, there are concentric layers of pus, this caseous pus that is so characteristic of reptiles. And if we have a look at a histological section of that particular lesion, we can see that in more detail. We've obviously got hyperkeratinization here, probably thickening of the epithelium. And then if we look at the dermis here, we've got first of all a caseous nodule. This is pus. This is a mixture of degenerate heterophils and other inflammatory cells, and on a different stain would illustrate mixed bacterial colonies. But in addition, there's vasodilatation in this area, there's edema, there's a fibrous capsule, and there is a moderate to marked mixed inflammatory infiltrate, again both granulocytes and mononuclear cells. So that's fairly characteristic of an abscess, a skin abscess in a reptile, and as I say, these are usually associated with a mixed bacterial flora. There are various other organisms that can cause infectious lesions of the skin in reptiles. Fungi, for example. This is a biopsy from a water snake, and this is a PAS stain, and you can see fungal hyphae here, nice magenta stained mat of fungus. Fungal infections of reptiles very often occur in captivity when the environment is not optimal, particularly, of course, when the relative humidity is too high, or perhaps when the substrate is unsatisfactory. And it's not at all uncommon for the species involved to be, as in this case, aquatic species where those environmental factors have been less than adequate. Now, suboptimum environment can also lead to what is probably the second most widely recognized infectious skin disease of reptiles, and that is so-called scale rot. Herpetologists call it scale rot. We would call it necrotic dermatitis or perhaps ulcerative dermatitis. What happens in this case is that because the environment is inadequate, changes can occur, perhaps associated with too high a relative humidity, too low a relative humidity, wrong sort of substrate, etc. Here, for example, we have a section through the skin of a snake. This is a scale. And you can see that most of this is normal, and certainly you've got the normal pigmentation, you've got melanin here, the dermis is relatively normal. But what we have got is changes here in the epidermis. We've got these indication of some changes, degeneration of keratin, and also some erosion taking place through the epidermis and almost reaching the dermis underneath. And this appears to be an environmental, non-infectious factor that causes this, as I say. But if prompt attention isn't paid to it, then it can develop, and one can get this sort of picture. And here we have a full-blown necrotic dermatitis. Another snake, but nevertheless a similar sort of picture. Here is the scale, but you can see that there is just the normal architecture has disappeared. There's just masses of necrotic material here, lots and lots of bacteria, inflammatory cells, etc. Very, very marked vasodilatation, inflammatory infiltrate going right through the body wall, through the muscle, and in this particular case resulted in a bacteremia, a septicemia, and the death of a snake. So what starts as a fairly minor skin lesion can very rapidly develop into this sort of life-threatening situation, and it's a reason why we must take it very seriously. I mentioned at the beginning of the lecture that skin lesions of reptiles can be proliferative, and you've seen some examples of proliferation in the context of response to insults and healing and so on. But neoplasms are increasingly being recognized in reptiles and should be mentioned in this lecture. To those of us who come from Europe, the most famous, if you like, and in many respects the most frequently seen neoplasm is this one. This is a lacertid lizard, Lacerta viridis, which is a European species, found not in Britain but out throughout much of the continent. And these have been well recognized as being host to a papillomatosis, And you can see two papillomata here, which are particularly prevalent around the base of the tail and around the head, and which can cause quite severe problems when these lizards are kept in captivity, particularly for research purposes. The appearance of this particular lesion on histology, this is a biopsy, is quite typical. You can see that the papillomatous structure here, the proliferation of the stratified squamous epithelium, quite normal, but with few, if any, mitotic figures, but nevertheless clearly papillomatous in structure. You can see pigmentation here, two types of pigment, and you can see part of the overlying epithelium, but that's not a particularly good part of the section. This particular papillomatosis is associated with a virus. Two teams, one in France and my colleagues and I in London 10 years ago, were able to demonstrate the transmissibility of the agent, and we were able to detect a virus as yet untyped on electron microscopy. So that is a transmissible neoplasm that is well recognized on our side of the Atlantic. But there are many other neoplasms of reptiles that are increasingly being recognized. I put this clinical picture in of a fibrosarcoma being treated in a corn snake, in an Elaphigata corn snake. One, to introduce the fact that fibrosarcomas are probably the most prevalent neoplasms seen regularly in captive reptiles, but also to make the point, of course, that as far as the pathologist is concerned, this is the worst way of dealing with the lesion. This is cryotherapy, and of course this renders the sample pretty well useless for decent histopathological examination. So we should make sure as pathologists that our clinical colleagues are initially removing a biopsy, or perhaps excising the lesion in toto, so that we have a chance to have a look at it histologically or perhaps cytologically or both prior to its being damaged by cryotherapy. If we have a look at in this particular case, at the section of that particular lesion, you can see that this is a very characteristic fibrosarcoma. You can see mitotic figures. You can see a lot of cellular pleomorphism. It's a poorly differentiated fibrosarcoma and a fairly typical picture, the sort of thing that one would see fairly regularly in this sort of case. Other sarcomas involving the skin have been reported in reptiles on a number of occasions, and the number of reporting seems to go up, possibly because reptiles are living longer in captivity and people are more conscious of them. This, for example, is a neurilemoma, a case that we published myself with colleagues here in the United States about 10 years ago from a Mediterranean tortoise, another sarcoma with rather different morphological features and cytological features, of course, which took some time to identify and characterized. We see other tumours. We see, these are sarcomas, of course, but we also see carcinomas in reptiles involving the skin, and some of these may be of relevance to captive breeding and conservation programs. Here, for example, a post-mortem specimen is what was one of the world's rarest species of reptile. This is the round island skink, which was confined to a very small island off the island of Mauritius in the Indian Ocean. And this was the species that the Jersey Wildlife Preservation Trust, or one of the species, that it saved from extinction by its captive breeding program. We encountered a number of problems in these captive reptiles at Jersey, and the one of relevance to this lecture is squamous cell carcinomas, which affected the feet. And you can see this particular lizard has got a lesion which each of them proved to be a squamous cell carcinoma on both a forefoot and a hindfoot. Time doesn't permit me to discuss what we thought was the underlying causation, the etiology of this, but nevertheless the important point is that we have to think of these neoplasms, including those that involve the skin, as being potentially significant in captive breeding and release programs, because many of these lizards were subsequently released and returned to the wild, and it's a possibility that this could be important in the wild as well as in captivity. Now, the ability of reptiles, particularly snakes, to shed their skins is a very, very important feature that has long been recognized and is very relevant to our interest in reptile skin pathology. And this is illustrated by This advertisement, for example, from a British company who are promoting their wares by saying the next best thing to a new skin, and they've put in a shed skin from a boomslang, a southern African snake here, is their particular product. It illustrates how this ability of snakes in particular to shed their skins has been long recognized and marveled at. And indeed, the inclusion of a serpent in the emblem for the medical and veterinary professions, one of the many reasons given for that is the ability of snakes to shed their skin and to take on a new healthy life when the skin is gone. That's probably overdoing it, but nevertheless there is a certain amount of truth in that particular statement, that losing the skin does provide a chance to free the reptile of certain diseases and pathogens and so on. Now, some reptiles will shed their skins or slough, as herpetologists in particular would say. Some will do it in pieces. For example, this East African chameleon is losing bits of skin. You can see the new, where the skin has been sloughed here, but there's areas of translucent skin, a piece that's come off here, etc., etc. That's fairly typical of a lizard shedding its skin, and very often lizards will eat pieces of that material, presumably as some sort of defense mechanism, and also the keratin may be of some nutritional value. In the case of snakes, however, the vast majority of snakes will shed their skin complete. And an early sign of this in snakes is the sign that you see here, whereby a translucency is seen of the spectacles. I'd remind you that the spectacles are the modified scales over the eyes of snakes. Snakes have no eyelids, they can't blink, whereas lizards and chilonians and the tuatara can, and crocodilians. And in this case, however, we are dealing with what are usually transparent shields, the spectacles over the eyes. At the time when shedding, sloughing is imminent, these become opaque because of, or translucent or opaque, because of an infiltration of leukocytes and various other changes and edema fluid, etc. And this is usually a sign that the snake is about to shed its skin. The distension that you see here is not normal, or is not normally seen. It was not, in fact, pathological in this case, but it does lead to the possibility of other differential diagnoses. But nevertheless, the picture illustrates well both that and also the change in the skin. The skin loses its normal sheen, etc., prior to sloughing. Now, the shedding of the skin, the sloughing of the skin, is a key part of the biology, the natural history of the reptile. And a good herpetologist, anyone who's working with reptiles, will record when the skin is shed, because this can be part of monitoring the health of that particular individual. And when things go wrong, that is when the clinician and the pathologist become involved. The term we use for difficulty in shedding the skin is dysectisis. Ecdysis, of course, means the change of skin, whether it's an insect or a spider or an amphibian or a reptile. Dysectisis is when there is difficulty in shedding the skin, or the skin shedding is abnormal. An example of that is shown in my next slide. This is a post-mortem specimen. This particular snake has obviously had difficulty in ridding itself of the old skin, and in fact we do have a clue here as to the cause in this particular case, because this snake also has these lesions, these ventral lesions, what herpetologists call blister disease. It is indeed a vesicular lesion in the skin there, in the superficial layers of the skin, and that is, blister disease is usually associated again with an adverse environment, particularly an adverse relative humidity. And this illustrates the fact that difficulty in shedding the skin is very, very often associated with underlying skin disease, and why that easily recognized clinical sign should be cause for investigation by the veterinary pathologist. If we look at a section of a snake that has not been able to shed its skin properly, we can see exactly what you saw macroscopically in the previous picture. We have here muscle, of course, we have dermis here, epidermis here, and part of a scale. And here we just have layer after layer after layer of unshed material, the outer layers of the stratum corneum, effectively, which have not been shed. And this is just built up and built up and built up. And this, of course, is not only per se not a satisfactory situation, but it can have secondary effects, and one that I'm particularly conscious of and teach is the fact you can get these little plugs of keratin here, and not easy to see at this magnification, but there are bacteria, mixed bacteria growing there, so the retention of a slough skin can lead very easily to a superficial dermatitis. Now, as far as dysectisis is concerned, it is a very good example of where the clinician and the pathologist have got to work together. It's a clinical problem, the reptile that doesn't shed its skin properly, but equally the pathologist can bring to bear his or her knowledge in order to help elucidate the cause. And when we run courses in Europe on reptile diseases, on comparative pathology, etc., we usually include a session on this. And in this picture you see two colleagues, one from France and one from the United Kingdom, who are actually looking at the shed skins of snakes, looking first of all with the naked eye for lesions to see if there's any evidence of old fibrotic lesions, etc., or areas of which are not normal, and then subsequently these slough skins will be examined with a hand lens or a dissecting microscope, and then pieces can be taken for culture. One very important tip here is that when these shed skins are collected, as any good collection should do, when a snake sheds its skin, it should be recorded and the skin is collected. It should be put into a sealed plastic bag and labelled. And that is not only because you want the sample to be properly collated and catalogued, etc., but very often there will be ectoparasites on the sample. And indeed, I often say to my students that the plastic bag is as important as the skin. You take the skin out and then hold the plastic bag up to the light. There may well be those little Ophionicis mites, for example, in the bag, and those can be a key to the underlying problem, the cause of the skin lesion. The inability to shed the skin properly is multifactorial. There's a whole range of factors that have been implicated. There are two broad divisions. There are the snakes in particular that have difficulty in actually removing the shed skin, and that is usually an environmental factor, and by raising the relative humidity or even soaking the snake in warm water, that will very often be corrected, and then of course in turn the management has to be changed. The other more complex form of dysectisis is the sort that you saw in that histological section with those layers of keratin on the outside, layers of shed skin, where it's a cyclical phenomenon, and almost certainly it's hormonal, endocrinological in etiology. And there's quite strong evidence that a hypothyroidism, either primary or secondary, can contribute to that. But this is a field that needs a lot more research. So it means that we have to leave no stone unturned in investigating the possible causes of this particular condition. Very often, as I say, environment is at fault. A collection where, for example, a tropical rainforest species such as a baboon viper is kept on sand is heading for trouble, and skin lesions and dysectisis will almost certainly be the first sign of that. And the same goes for poor hygiene. Here's a collection in a part of a collection in East Africa, an African rock python, where the conditions are poor, the hygiene is very poor, there's urate material, there's bits of shed skin, etc. And this snake had a low-grade dermatitis, which was contributing to its inability to slough properly. So those are just examples of the sort of factors that can contribute to this. So the environment is all important, and we should be visiting when the opportunity arises, collections, zoological parks, captive breeding programs, conservation programs, and seeing what sort of accommodation is provided, what sort of substrate, what sort of temperatures, etc. And I believe this to be a role for the pathologist, not just the clinician. But it is nevertheless very important not only to look at the environment, careful clinical examination of the patient is also necessary, and again I would argue that the pathologist should play a part in this. It's a great help working in Africa, where I do frequently, to have lots of students, because your patient may be a long rock python, as in this case, and this is the time when the snake can be properly examined. ectoparasites removed, biopsies taken, cytological preparations, etc. So the pathologist, in my view, must play a part in the clinical examination and in the taking of samples, not just in the pathological, in the examination of samples in the lab when they've been taken by someone else. I'd like to conclude this lecture with a little poem. And the poem is written by a medical practitioner, and he's describing here what he sees as the exquisite features of the skin. And it reads as follows: "It covers every inch of us from crown of head to toe, and through each tiny fragment the busy red cells flow. It's living, pulsing, breathing, percipiently sensing, one moment lying slack relaxed, another tautly tensing. It holds at bay bacterial germs, it keeps our innards in, that wondrous complex structure that is stratified squamous skin." Well, that of course refers to human mammalian skin, and if I conclude with a picture of a mammal, a rock hyrax in the Serengeti National Park on the right, and a reptile, an agama, a lovely male agama with multiple colours on the left there. It's important to remember these are both members of the same phylum. They're both chordates, but we have the class Mammalia on the right, the class Reptilia on the left. There are many differences, and I've emphasized those, but there are also many similarities. And the key similarity relates to the poem, and that is that the skin is the largest organ of the body, and a very, very important one in terms of our detection of disease and indeed our understanding of comparative pathology. Thank you.